Thank you so much for the introduction, Antonia. Yes, so I am working here in Oxford in the Department of Biology. Supervisor I Professor Craig McLean and Mike Brokos. So hopefully the talk is going to be interesting for you. I remember one of the guys from Imperial College London were asking about if we were interested in exploring the interaction and evolution between the phage and the host. So we're actually quite interested in what we actually do. So I will try to let you know about the potential evolutionary outcomes of the interaction between the phage and the host. In this case, is more interesting for me at least because it's the interaction between the phage and the target that the phage target, which is another mobile genetic element, broad host range plasmid. So just quickly go to the big issue that we have currently, antimicrobial resistance. So antibiotics were discovered almost 80 years ago. They were like the gold option to save people from bacterial infections. So before that, we had like very high mortality rates. So that was like the hero that we have. However, because we abuse a little bit of the use of antibiotics, doing like basically using them for even as a growth promoters and also over doing over prescription of antibiotics in hospitals. So basically what we did is these high toxic molecules impose a strong selection pressure in populations, in bacterial populations. So we select for the most rare mutants in the population. So we move forward from this quite level of antimicrobial resistance to a high frequency of those genotypes in the population. So we are moving forward basically from just 80 years ago to the current situation from a pre-antibiotic era towards a post-antibiotic era, but basically antibiotics are not able to kill bacterial pathogens. So yes, this is going worse because basically we are not producing new antibiotics. Bacteria already found a lot of solutions to survive against these antimicrobials. And so basically we enrich for these very successful bacteria that can overcome the commensal bacteria in host. So these superbugs are making a lot of troubles, and we are a lot of people is dying for the infection of these bacterial pathogens, especially from the escape group. So before discovering how antimicrobial resistance was transferred and shared between bacteria, we know that the only mechanism to acquire resistance was basically by mutations. So however, in the 50s, after the discovery of the plasmid and this behave that we found in bacteria, that they are able to share a small molecule that we name plasmids, and we analog that bacterial behavior with human sex or sex overall in animals, that basically is an higher rate of recombination between organisms. So we found that actually the same factor that were shared by bacteria by this mechanism that we know is conjugation are the same that carry usually antimicrobial resistant genes. And that is because those genetic elements basically are the molecules that bacteria use to share adaptive traits like antimicrobial resistant genes. So they are pretty good sharing the bacteria, sharing these genes. So basically conjugation is a self-dependent contact mechanism that is leading by basically an appendage that is basically a bridge that the bacteria use to transfer DNA, including these plasmids. So the plasmid is basically the one that confer and encodes all the genes that are necessary for this mechanism of DNA translocation. So how antimicrobial resistance is spread across microbial communities is basically this is a picture that shows how the mechanism goes. So it was a very nice picture, a very old one, showing the conjugation in F plasmids. So basically the bacterial donor, the bacteria that carry the plasmid, is able to transfer the plasmid to cells when they are coexisting. Basically, they move from bacterial populations to microbial communities. Microbial communities coexist with other microbial communities in bacterial hosts, like basically between microbiomes. And then basically we are moving those genes between different microbiomes. So that is the most successful mechanism to share and evolve antimicrobial resistance. And also to acquire different bacterial traits that are necessary for bacterial survival rates, depends on the stress that is imposed by the environment, right? So in this case, we are just focused on the antimicrobial resistance, and it's something that actually is introduced mainly by humans. So the phage potential that we believe, so we are looking for alternatives. Everyone already know that. We are approaching phages as an alternative. However, how we can target this very common and successful mechanism to mobilize AMR genes. So conjugation is, as I say, is a mechanism, and there are some phages that target actually conjugate pili, as I mentioned, is the main structure that is the responsible for the mobilization of plasmids. And so the plan is basically using phages that can counteract the role of plasmid in horizontal gene transfer. So this kind of donor-specific phages, pili-dependent phages, sex pili-dependent phages, or PDPs, how usually we start to name them, that is plasmid-dependent phages, are quite abundant in nature, however, are a bit hard to isolate. So yes, the plan is blocking the conjugation mechanism. So the phage that we are using as a model to understand the interaction is PRD1, or brilliant PhD student just talk you a little bit about this phage and their potential to block conjugation. So, but we really want to understand the evolutionary outcomes of this interaction. So that's the reason that we are using experimental evolution approaches. And so PRD1 has some, in general, phages from this group have this feature that they are, the host range is limited just by the host range of the plasmid. So actually they can infect different species. And what the phage target is actually is a quite broad host range plasmid. So it's the plasmid that we are, so the host, the plasmid host that the phage target is INPI plasmid mainly. There are also other plasmid that can be targeted, but the phage is quite smart because it's targeting one of the plasmid with the broadest host range. So before these plasmids were studied because their ability to transfer genes to any kind of bacteria, including gram-negative, gram-positive, like Staphylococcus aureus. However, that probably doesn't happen frequently in nature. So yes, PRD1 target INPI plasmid, as we can see, the phage can kill Pseudomonas or E. coli, and it's a non-phage phage. It's a non-tailed phage that is unusual in phages. However, makes sense why they don't have a tail. Later on, I will try to explain you why. So INPI plasmid, as I mentioned, are one of the most successful mobile genetic elements in nature. They can spread across gamma perturbacteria. So those are the main groups that usually we can find them: Pseudomonas, Enterobacteriaceae, different species like Escherichia, Klebsiella, other bacteria that host this kind of plasmids is Burkholderia. Anyway, basically the bacteria that we consider are like the human enemies, pathogens. And these plasmids are usually are conjugative, so they can mobilize. That's the reason that the phage target this kind of plasmids. And they usually carry more than one antimicrobial resistant gene. So definitely they are AMR spreaders, a broad host range plasmids. So how we explore this interaction? So basically, we do experimental evolution. We follow the dynamics of the different population that we are expecting that the phage the population that the phage interact directly or indirectly. So you just follow the density of the population, and after that, we try to characterize just the evolutionary outcomes, basically the resistance and the phage. However, basically, just I want to mention that the plasmid that we use is RP4, as I already said, but it was modified. So we introduced a marker, a fluorescent marker, GFP, to make easier the following of the mobilization of the plasmid towards the recipient cell. That is another E. coli that has a chromosomal resistant gene that encodes resistant to sodium meside, J53. And basically, we're following the density of the events when the recipient acquired the plasmid, and also the phage density, as I already mentioned. So we use high throughput sequencing to study the evolutionary outcomes. So what's happened after interaction? So we have two conditions: no phage and PDP, that is basically PRD1, the phage that we use. And these are the colors indicate the population that was the density of the population during the time that we follow the the interaction. So in this case, these are days. And so we keep following the dynamics of the population until we notice that the system was not still moving as we were expecting. So basically what we can observe is when we don't have the phage, the recipient cells that are in blue, they can acquire the plasmid from the donors and then can reach a very high density and basically they just reach the same density than the bacterial donor. So that means that the plasmid is quite successful and spread in a very high density in the recipient population. The transconjugates, sorry, the transconjugants were the purple. So the recipients are the cell that actually they then acquire the plasmid. So usually the acquisition of the plasmid is just like 1 order or 2 orders lower than the original host of the plasmid. To make this more interesting, we decide to introduce more recipients because we know that basically the purpose of the plasmid is a parasite and they want to spread in more cells. So we introduce more cells to trigger the expression of the conjugative pili and then to basically increase the conjugative rates. So we introduce more cells and basically what we observe is that the plasmid is still acquired and at very high rates. So what's happened? You add the phage. So basically in green, we have the phage is the new variable that was not in that condition. And so the first days, basically the phage increased importantly. The density is like a burst of almost 100 fold. And then you have an important killing effect in the donor population. That is what the original population carrying the plasmid. And as you can see, we have an important suppression effect in the acquisition of the plasmid. So the purple line that represented the acquisition of the plasmid cells, the transconjugates, it was reduced importantly. So this was happening for the whole time of the experiments. We observed some interesting dynamics when the plasmid start to be acquiring a little bit more, and that is correlated with the decrease of the phage population. So that suggests for us that basically resistance was appearing in the population. And so basically the phage was not able to replicate. And then that's the reason that also the original population of the carrying the plasmid, the donors, start to recover the density. So that's happened in a similar way when we try to trigger the mobility of the plasmid. So the phage was maybe able to survive for longer. So that's mean that potentially the introduction of more recipient cells help to the phage survive for longer time. So probably in nature, this is a more realistic model when you have like more potential host for the plasmid. So then to really understand what's happened in the system, so first, basically this is like just assume to the population that we were expecting that the phage will affect. Basically, this is the density of the donor and the transconjugate. As you can see, the phage